Hi, and welcome back to the Reconciliation Project podcast, where we are formed by faith and informed by science. I'm Sam.
And I'm Hunter.
And today we're going to be covering evolution and genetics. So specifically, how does genetics play a part in natural selection, which is what was the main mechanism that Darwin used as the basis for his theory of evolution. So you actually are completing a master's in genetics, so you know a lot more about what we're going to be covering today. Of course, we're going to be covering history of it first. So do you want to jump right off and get into that for us?
Yeah, so we know that genetics as a whole kind of really started with Gregor Mendel when he did his pea plant study. And he was doing that, as we talked about, I believe, in the previous episode, where he was doing this right around the same time that Darwin was actually doing his studies in the Galapagos Islands. And basically what Gregor Mendel was doing was he was studying how traits are passed down through generations. And he did this in pea plants. For anyone who's gone through any level of high school biology, you've probably heard of Gregor Mendel and his pea plant studies, but you may not be familiar with exactly what he did. Basically, like I said, he was trying to study inheritance patterns, and he was looking at a number of different traits, namely seed color, seed shape, and plant color, are like the big three that we're often taught about. And he looked at each of these traits and saw how they were passed down. Because originally it was thought that a person throughout their lifetime, they acquired trait and then they passed those traits down to their offspring. Like we talked about with Lamarckian evolution with the giraffes, they stretched their neck, they would pass that down. However, Gregor Mendel, he kind of challenged that idea when he discovered that traits weren't passed down based on what was acquired, but they were passed down by something else inside. As a matter of fact, when he first crossed, talk about plant color, when he first crossed a purple plant and a white plant, he found in his first generation that 100% of the flowers in that first generation offspring were all purple, which is surprising if your idea is traits are just passed down based on, I guess, outward appearance. But he discovered that all of them came out purple, and he crossed two members of that generation again, and he found roughly 75% of the flowers were purple and 25% of the flowers were white. And as he continued doing this, he found these patterns to repeat. The first generation, only one of the two traits actually appeared, and it took 2 generations for that other trait to appear again. And what he found with this was dominant and recessive traits, or dominant and recessive alleles. And what I mean by allele is, basic definition, is alternative forms of a gene. And basically, if we know, as humans, we have 46 chromosomes, 23 pairs, and we get one set from each parent. We get one set of alleles from one parent and one set of alleles from another.
So each gene is made up of 2 alleles for the same gene.
For the most part, yeah. Some genes actually have more alleles than others, but
for example, blood type.
Yeah, but at the most basic levels, most genes, or some genes, have 2 alleles coding for them. In the case with the flower color, there were 2 alleles. There was a dominant purple allele and a recessive white allele. What I mean by dominant and recessive is if the dominant allele is present, the recessive allele is completely blocked. It doesn't appear. So as what Gregor Mendel found, the white recessive allele was completely not there. It was completely covered by the purple trait. It's only when 2 copies of the recessive trait are passed on that that trait actually appears in the person as a whole.
And this is because if the dominant allele isn't there, there's nothing to cover the recessive allele. And therefore, the recessive allele can then show through in whatever that shows up as in a physical trait.
Yeah. So the only way for a recessive trait to appear is for there to be 2 copies of it in a person's DNA, or in this case, in the pea plant DNA. For a dominant trait, all that's needed is 1 copy. And overall, what this yielded was the idea that there was something else causing traits to be passed down, that something else was carrying information. Now, at the time, he didn't know anything about DNA. It wasn't until the 1900s that the idea of DNA being the carrier of genetic information and the double helix and all those things coming about. That didn't happen until the 1900s. So Gregor Mendel had no idea about that. He just knew that something else was responsible for passing traits down. But later on, as the field of genetics started to emerge and molecular biology started to emerge, we found that DNA was in fact the genetic material. In other words, DNA is what makes us who we are. And molecular biology, and also Mendel had parts of this as well. As a whole, they came up with what's called the central dogma, which essentially states that DNA codes for RNA, which codes for protein. So it's ultimately all the genetic information goes back to the DNA. Now, there are times where the central dogma isn't absolute, especially now as we've continued molecular biology studies. We know the central dogma isn't an absolute. It generally holds up, but there are cases where we see DNA being made from RNA. And this is specifically how retroviruses like HIV work. They pass their RNA-based contents into a cell, and that cell then makes DNA from that RNA, and the cell essentially becomes a virus factory once it's been essentially hijacked. So there are cases where it can go the other way or doesn't go in that one linear direction, DNA, RNA, protein, as Gregor Mendel or other early molecular biologists would have originally said. However, the overall point of the central dogma does hold in general. We know that DNA has to get replicated for cells to grow and divide, because cells, they go through a process called mitosis, where the cell essentially splits into two identical cells. But before it can do that, the cell has to duplicate its organelles, or the small machines within the cell that makes it work. And it also has to replicate its DNA, because if it doesn't do those things, the daughter cells, the resulting cells from mitosis, won't work, or they'll become cancerous, or they'll just die.
Absolutely. Each cell needs a full working set of DNA. In humans, needs a full 46 chromosome set.
So replication occurs to make an exact copy of the DNA from the original DNA. Basically, what this means is the process of DNA replication is called semi-conservative, meaning it uses the existing DNA as the template to make an additional copy for the future daughter cells. And the point of this, as you point out, as we've mentioned, is to ensure that the two cells resulting from that original cell are able to function normally. And when this process goes wrong is when we can have things like cancer popping up. I would encourage anybody who's remotely interested to go look up on Google a cancer karyotype. And the reason I'm saying that is because if you look at the genetic information found in a cancer cell, it's really interesting and also really scary is how messed up the DNA in a cancer cell can get, simply from issues with replication that don't get checked, or other issues with mitosis that happen because these cells aren't regulated as they should be. And the point of this regulation is, as we've mentioned, to make sure the cells work correctly. But we get from DNA to DNA, but how do we get from DNA to RNA and then RNA to protein? Well, we have another process called transcription, which is very similar to replication in that proteins will run along the DNA to make an RNA copy. Now, the difference between DNA and RNA, as a general rule, DNA is double-stranded and contains 4 bases, an A, T, C, and G, whereas RNA is generally single-stranded, not always, but generally, and it contains the bases A, U, C, and G. And RNA acts as the messenger from the DNA genetic code to create a protein. In humans and eukaryotes as a whole, the RNA has to get processed. We're not going to go into that really much, but the RNA, when it gets copied, has to get processed before it can go make a protein. And what does that? A lot of protein. Or before finally where we get to what's called a ribosome in the cell, where translation happens. And this is essentially where the RNA strand is read and from that the ribosome builds a protein. So overall, the idea is the DNA is required to make RNA. RNA is required to make a protein. However, proteins are required to replicate DNA. Proteins are required to transcribe the DNA into RNA. And yet more proteins are needed and more RNA is needed to build proteins. So it's kind of a circle here. They all need each other. The DNA needs the RNA. The DNA needs the protein. Proteins need RNA and proteins need DNA. Like I said, they all need each other. So this really creates an even more complex chicken and the egg situation. We've all heard what came first, the chicken or the egg?
It's very similar with this, especially when we're coming back to the evolution component of this episode. Evolution-wise, you make the argument that, again, back to what we mentioned in our last episode, you need to have successive steps that build to a greater complex system. But if DNA needs RNA and RNA needs proteins and so on and so forth, and they're all kind of tied together and you can't tell which one came first, there's the very real argument that evolution could not have worked because the process for creating DNA, RNA, or proteins wouldn't have worked in those successive steps.
And currently there is the idea that the early Earth was made of, I guess I should say, contained RNA first. That RNA arose first, because RNA as a whole is a more simple structure. And the idea is that from RNA, we got proteins and DNA. And as I mentioned before, there are cases where RNA can be used as a template to create DNA. However, this still requires specific protein to happen. And even so, RNA, going back to DNA, is not a 100% occurrence across the entire animal kingdom and across all of life. It only happens in certain organisms, in certain cases that actually need RNA to go backwards into DNA.
For example, back to your virus example that you gave, according to current taxonomy, viruses aren't considered to be alive. So the question is, how do we go from a virus, which isn't considered to be living, to maybe a bacterial cell that is considered to be living?
So for sure, and it does create a challenge for explaining how early life arose. And there are other cases, I saw this recently, that they actually discovered that certain bacteria can use their protein to create DNA as a way of creating sort of an immunity or a defense mechanism against viruses. And this is something that's, like I said, pretty new, but it is further shaking this central dogma being absolute. And it's really giving us a whole different perspective on genetics as a whole, that it may not at all go in one direction, but it can go all multiple different directions. And we're going to get into this a little later, but we didn't even mention the regulation and the other things that are involved in making sure the transcription translation processes happen correctly as well. Those also get heavily regulated. So overall, I bring up the ability of some bacteria to be able to create DNA from a protein. Very specific to those bacteria, as far as we know right now. But it does, for one, bring up a potential mechanism for evolution, and it does further challenge the central dogma as an absolute.
Now, absolutely, this isn't necessarily saying that, oh, then evolution has to be real or evolution doesn't work at all. We mentioned earlier that microevolution does happen with speciation. We see it happening on small scales. We're not trying to completely jump one way or the other on an extreme. It all comes down to having more evidence that we can use in our line of reasoning, but that also comes with the argument that, hey, we still have no idea what's going on here. We're discovering cool things, but we have no idea how this works, how it came to be this way. We have no idea. And it requires a lot of further study.
It does, for sure. And as we continue studying into bacteria, because bacteria have a lot of really fascinating things that they can do with their genetic material to defend themselves against viruses. I mean, you think the smallest, most simple forms of life have these really intricate mechanisms that allow them to survive, that help them to survive. And they develop these mechanisms over time as a way to further grow and further live. And I think it is reasonable to assume that if a bacteria can create DNA from a protein, well, for one, odds are this is using other proteins to do it. And odds are it's probably using some other factors as well that we don't know about. And it's also really doesn't provide any new information in terms of how life arose from non-life. Because even if you can get DNA from a protein in a primordial cell, you still have to be able to take care of that DNA. We know that DNA is very well kept in the nucleus of the cell to make sure it doesn't degrade. If DNA is left out, it can degrade. It's part of why in fields that need DNA to ever work, such as forensic DNA, a lot of times they find DNA that's degraded because it's exposed to the outside environment. And it typically doesn't take too long to happen, so it has to get really well kept or else it's not going to survive. So my point is, in a primordial Earth, the DNA, we would have to figure out how was the DNA kept stable in this situation without the nucleus, without any other things to keep it from falling apart. So while yes, you could say, well, these things arose by chance, just by what was there, you then have to talk about how did they stay together? How did they stay stable long enough? Because you don't get life simply from protein. You don't get life simply from RNA. You don't get life simply from DNA. You need all three and other things that come from these three to remotely begin having life.
Absolutely. Let's go back for a second to your comment about DNA degrading. Obviously, there's a lot that can go wrong when DNA is left out, but we also see a lot going wrong sometimes within cells, even when the DNA has been kept in the nucleus and in a safe environment. Do you want to elaborate a little more on kind of where DNA can go wrong?
Yeah, for sure. That leads us into talking about mutations. And one of the big ones, I'm just going to jump down here, when talking about the DNA, even when it is kept well in the cell, it's still vulnerable to damage. One thing that I think we're all familiar with, especially now that it's summertime, that it's hot outside, people are spending time outside. We know that UV radiation from sunlight can damage the DNA. And what it does is it creates what are called dimers, which is essentially when two of the bases in the DNA structure sort of bond to each other, creating like almost like a dent or a kink in the DNA backbone. And while this doesn't create issues initially, we go back to replication. When the DNA is getting replicated and the proteins that are responsible for creating the new DNA strand run into these dimers or these kinks in the DNA backbone, they can't go any further. They do one of three things in these cases. Either it'll stop and fall off, it'll skip the spot entirely, or it'll put some random base on the new strand. And what this does is creates a mutation. And in these cases where UV radiation becomes deadly is when the radiation actually can break the DNA strand as a whole. And double strand breaks are, in humans especially, not that easy to repair. And the main reason for this is because double strand break repair in mammals, humans are mammals, so in humans, double strand break repair is not the best process. It poses issues. It's not 100% good. It doesn't work 100% of the time. Typically, because of the mechanism for double strand break repair works in humans, it tends to create more mutation. And as you create more mutations, and this happens over and over, eventually you either kill the cell or you lead to what is arguably the worst of the two results. You get cancer. So you can see where mutations can cause problems. But there are other types of mutations. There's point mutations, which are simply just a change in one base in the DNA. Remember I talked about the bases A, T, C, and G. So one of those gets changed. There's what's called a frame shift mutation, and that's when a base is inserted or deleted and it moves the reading frame. And by reading frame, I mean that's what provides the information for creating protein. And when a multiple of other than 3 gets inserted or deleted in the DNA strand, it creates a lot of problems for the future protein. There's silent mutations, which are simply just changes in the DNA that don't affect the protein. There's missense mutations, which are going back to point mutation and frame shift mutation. These are mutations that create some sort of change to the protein structure. And then there's nonsense mutations, which when these occur, they can actually cause the protein structure to be shorter than they're actually supposed to be.
Now, obviously, a lot of the consequences you just mentioned are mostly harmful. You mentioned silent mutations being those that don't really cause any real change, and those are more neutral. But there are also other options for kind of those neutral mutations as well, mostly when they occur in those regions that are regulatory, that regulate how the DNA is read, how the protein is formed, what have you.
Yeah, typically, we know most mutations are neutral, as you mentioned. The human genome can experience thousands of mutations in a person's lifetime. And this shows how usually a mutation doesn't really have that much of an effect. And typically, these are in those non-coding regions of the DNA that don't necessarily affect the protein itself. So as a whole, mutations are usually just neutral. They don't really do anything. But as we mentioned, they can be harmful as well. And one of the things that comes up in evolution discussion is the idea of beneficial mutation. And the idea of this is, can a mutation that happens create actually a benefit for the organism? And there are times where this can seem that way. However, as a whole, mutations really never add anything to the DNA. They never add anything to the genome. And really, they're only beneficial in cases where the environment actually makes that possible. For example, when a person has sickle cell, their red blood cells are in a sickle shape, and there's a rigid, hard structure that can't carry oxygen as well as the disc shape that is normal. However, when a person has only one of the two, going back to what we talked about with alleles, if they have one sickle cell allele and just one regular allele, these people, because they have that regular allele, they still are able to live fine. They aren't considered to have sickle cell because they have red blood cells that are the correct shape, and thus they have oxygen flowing normally. But because they have that sickle shape on the other side, they're actually less susceptible to malaria in regions of the world where malaria is very rampant, such as in Africa.
Exactly. The other side of those people who have both the sickle cell and the non-sickle cell alleles is they are what we now consider to be called have sickle cell syndrome, which comes with the respiratory distress of sickle cell, not being able to breathe as well, having some episodes here and there, depending on how bad your situation is. But they don't have the full-blown sickle cell anemia. So while having kind of the halfway of one allele and the other does help in cases of malaria, it also has drawbacks.
It does. And in cases where malaria isn't really an issue, such as in the US, you really have no benefit from that. So at the very least, it's neutral. So my point is, while they may seem beneficial, it's really beneficial based on the environment. And that is where evolution does come into play, where the trait does get passed on in a certain environment. Again, going back to natural selection. However, as a whole, there aren't many mutations that we could consider calling beneficial. And I would argue that there's really no such thing as beneficial mutations as a whole, simply because there's a lot of other factors that come in to these mutations. As you mentioned, even in those cases where a person has that benefit from having the sickle cell gene, they still have some drawbacks. In these cases where a person has a sickle cell gene, they might still have some issues. So overall, going back to evolution as a whole, the question is, how do mutations occur and how do they affect natural selection? We talked about how they occur. It can happen with issues with replication. It can happen because of the environment. It can happen because of things a person ingests or an animal ingests, a plant. Sometimes plants can take up toxins from the environment. So now how do they affect natural selection? Again, we've already talked about some with if a trait comes up and it provides a disadvantage for an organism in a certain environment, it's likely that that trait will eventually get selected out because they just are that disadvantaged in an environment. And that's why we see a lot of genetic disorders that are harmful. They're pretty rare because a person typically can't live that long with some of them we know a person can't live that long with, and so they never reach reproductive age, therefore that never actually gets passed on.
Mm-hmm. So overall, mutations relate to natural selection because mutations are changes in the genetic code, and the genetic code affects your physical characteristics, which is then where natural selection can act on.
Natural selection isn't going to select the DNA itself. It selects the results of the DNA, which we call the phenotype. If a certain phenotype is good in the environment, it's going to be selected for, versus if a phenotype is bad, it's going to be selected again. Again, going back to the giraffes and the long neck. Longer neck phenotype got selected for because they had a competitive advantage over giraffes with a shorter neck. So overall, if a mutation is neutral, it doesn't really have much of it. Natural selection is probably not going to play one way or the other. If a mutation is harmful, odds are natural selection is going to select against it. If a mutation happens to provide some sort of benefit, natural selection may favor it. However, as with the sickle cell, we still know that the non-sickle cell trait is still most common, even in those cases where malaria is a factor. So as we transition into looking at how DNA has been used as evidence for evolution, common descent, we first need to look at what has been long known as junk DNA and pseudogenes. So junk DNA is an idea that arose from the fact that only 2% roughly of the human genome is actually responsible for coding for protein. And for a long time, the other 98% was considered to be remnants of millions of years of evolution, mutation, and natural selection. In other words, the rest of the genome was the results of common descent. The original ancestor had much more that was transcribed, and now we just have the 2% that we need, and the rest is just there from the results of evolution. And pseudogenes are a specific type of junk DNA that look like genes, like in terms of their code, they look like genes, but they're actually completely dysfunctional. However, new studies have shown that much of the human genome is transcribed into RNA, at least, or it's involved in the regulation of the genome, or it directly codes for genes. So rather than just that 2% that's responsible for genes, we actually know that much more of it is responsible for creating RNAs and regulating the genome, talking about regulating replication, regulating transcription, regulating translation. And as we've continued looking into these junk regions, quote unquote, we've actually found that there are several diseases that occur as a result of mutations in these quote unquote junk regions. For some examples, there's myotonic dystrophy, Fragile X syndrome, Friedreich's ataxia, and this one's a long one, facioscapulohumeral muscular dystrophy. 4 diseases that are specifically the result of mutations or errors in non-coding regions of the DNA, or what's been longly considered as junk. So what we know from this is these regions, if they result in diseases when they're mutated, they can't be junk, simply because if they were junk and in fact they didn't do anything, these diseases wouldn't happen.
Mm-hmm. And that's a huge argument against the evolutionary idea that, oh, these are just remnants from past evolutionary lines. If that were the case, they would be 100% correct to say, oh yeah, this is just junk, it doesn't code for anything, it's literally just a relic. But clearly it's not a relic, which casts a lot of doubt on the evolution component.
Yeah, and like I said, we're finding that a lot of the genome is responsible for just regulation. There are so many types of RNAs far beyond those required for just carrying bring information from the DNA to the ribosome for translation. We know there's some that's involved in building proteins, but even beyond that, there's so many involved in just regulation and other functions in the cell. And we know that a lot of the DNA actually codes for that. And there's also code or regulatory regions of the DNA that aren't transcribed, but they're used for showing, in case for the enzyme that's responsible for building DNA or creating RNA from a gene, there's some sequences in the DNA that essentially tell the protein, hey, the gene's down there, go that way. So we know there's a lot of sequences in the DNA that are actually responsible for regulation. And the overall point of this is it shows that even though for a long time it seemed like only 2% actually did something, we're finding that a lot of the genome actually has some sort of purpose. As I mentioned before, molecular biology is still a relatively new field, but as we continue on, we're discovering more and more. We're discovering more and more about the functions of these mysteries of the genome, for lack of a better term. And then lastly, another big piece of evidence used to support evolutionary ideas is the genetic similarities that humans have with other things. So we know that 98 to 99% of our DNA is actually shared with that of chimpanzees. We know that 85 to 90% of our DNA is shared with mice. We know that roughly 60% is shared between humans and chickens, and up to 60% is shared between humans and bananas. And that may seem surprising, but we've done sequencing of these organisms, we've done sequencing of the humans, and they're largely similar. And the question ultimately is, if evolution isn't a thing, then why do we have such similarity? And because that question arises, it does further support the idea of, or it's used as further support for the evolutionary theory from common descent. However, we know that as Christians, we believe that God would have likely used a very similar template across all of life. We know that an author, when they write a bunch of different books, you can tell the author based on their similar writing patterns, based on the verbiage they use and other factors. We can see these similarities across different works, even though they're completely unrelated, because they came from the same author. Same thing with art. You're bigger on art than I am. I think I'm accurate in saying that art, you can typically see similar themes across different pieces from the same person. And I think the same principle can be applied to biology and life. We have the same creator, and we're going to see similar themes across all of life, even when various parts of life are completely different from one another visually. And I think that's where we get these genetic similarities and these other similarities that we're going to talk about as we continue on with our studies of evolution. And I think that's a great place for us to wrap up.
Absolutely, yes. So thank you again for joining us for this episode of the Reconciliation Project podcast for our talk on how genetics and evolution kind of are tied together. As we continue with our discussion of evolution, we're also going to be discussing how evolution and embryology are tied, which embryology being how life is, as Christians would say, knit together in the womb. So we hope you will join us for that.